In this session, we will study the histology of different types of cartilages. Cartilage is specialized type of connective tissue and it is meant for bearing body weight. Then in case of long bones, we have epiphyseal plate where growth occurs. So it helps in growth process. Then it supports the developing organs. So cartilage is seen wherever rigidity and elasticity is required. Also cartilages help in withstanding bending and torsional forces. Peculiarities of cartilage are that the cartilage develops from mesenchyme and as we have seen the components of other connective tissue, the cartilage is also formed by cells, fibers and ground substance. So the cells are mainly chondroblast and chondrocytes. As we know, the chondroblast are mitotically active cells and chondrocytes are resting cells. Then the fibers are mainly collagen and elastic fibers, and the ground substance is formed by mucopolysaccharides, which includes chondroitin sulfate, keratin sulfate and hyaluronic acid. Then the cartilage is avascular and non-nervous tissue. It has anti-angiogenic factors which inhibit vascular invasion according to some authors. And as it is avascular, it receives nutrition by diffusion process. And the cartilage cells grow with two types, means there is oppositional growth and there is interstitial growth. So in oppositional growth, the cells are deposited beneath the perichondrium, so they increase the width of the cartilage, and the interstitial growth occurs within the matrix. So the chondrocytes proliferate by mitosis from the center of cartilaginous model, and this increases the length. So we have seen cartilages grow with appositional growth which increases the width and interstitial growth which increases the length. Then the cartilage is surrounded by a fibrovascular membrane known as perichondrium. So the perichondrium covers the cartilage except for the articular cartilage and fibrocartilage. In these two varieties, the perichondrium is absent. Then homogeneous transplantation without rejection occurs in case of cartilage because the cartilages have lower antigenicity of matrix, and also there is isolation of chondrocytes in the lacunae. Steps involved in the histogenesis of cartilage are depicted in this slide. So cartilage develops from embryonic mesenchymal cells, and the process of development of cartilage is known as chondrogenesis. So we can see the figure. So the mesenchymal cells are seen here. Then these mesenchymal cells retract their processes and they multiply and they form chondroblasts. So there is condensation of chondroblast as seen in the second step of that diagram. Then after synthesis and deposition of matrix, the chondroblasts are converted into chondrocytes. So in the third figure, we can see the central cells are chondrocytes and the peripheral cells are chondroblast, and the superficial mesenchymal cells develop into perichondrium. After the conversion of chondroblast into chondrocyte, these chondrocytes will secrete phosphorylase and it will convert glycogen to sugar phosphate. Then it secretes alkaline phosphatase and it converts sugar phosphate to free phosphate. And later on, it combines with calcium phosphate, and this entire process where the calcium is laid down in the tissue is known as calcification. Then the chondrocytes in the calcified matrix will die due to lack of nutrition by diffusion, and the osteoblast with blood vessels will deposit in the new bone of calcified matrix. So formation of new bone by osteoblast is known as osteogenesis, or we can say ossification. So there is difference between calcification and ossification. Calcification is simply laying down of calcium phosphate in the matrix, whereas formation of new bone is known as ossification. Cartilage is an avascular structure. So how does it receive nutrition? So the nutrition to a mature cartilage is derived from diffusion process. So diffusion occurs from the capillaries in connective tissue or by means of synovial fluid present in joint cavities. And in case of embryonic life, cartilage canals are present. So these are small canaliculi in which the blood vessels are present and they nourish the chondrocytes. And the cartilage canals also help in ossification of cartilage by carrying bone forming cells into it. And if there is nutritional deficiency, like if there is lack of vitamins and proteins, it results in diminished thickness of epiphyseal plate. So epiphyseal plate of the long bone is the part where the growth of bone occurs. So if there is lack of vitamins and proteins, there will be diminished thickening of epiphyseal plate. Then deficiency of vitamin C causes improper matrix formation, and lack of vitamin D results in excessive proliferation of cartilage with diminished ossification. So this will lead to deformities in the bone. There are two ways by which a cartilage grows. So first way is oppositional growth. As we know that a cartilage is covered by fibrovascular membrane known as perichondrium, and this perichondrium again has two layers. Outer is fibrous layer which is vascular, and inner is cellular layer. So the cellular layer has chondroblast in them. So when the chondroblast in the chondrogenic layer of perichondrium divide, and when they deposit new matrix, there will be growth of cartilage, and cartilage will increase in width. So such type of growth is known as oppositional growth, which mainly occurs due to the deposition of new matrix by chondroblast present in cellular layer of perichondrium. And the second method, or the second way of growth is interstitial growth. So we know that within the cellular matrix there is multiplication of chondrocytes. Means in the matrix there are lacunae, and within the lacunae there are chondrocytes which will divide, and they will again deposit new matrix, and this will increase the length of the cartilage. So when the chondrocytes present in the lacunae within the matrix will deposit new matrix, it will lead to increase in length of the cartilage. So it is known as interstitial growth. And both these types of growth, appositional and interstitial growth, can be seen in the epiphyseal plate of a growing long bone. And once the growth is completed, all the cartilage is replaced by bone tissue, and now the growth plate is completely ossified. So no more growth of the cartilage occurs once the growth plate is ossified. Cartilage is covered by fibrovascular membrane, which comprises of outer fibrous vascular connective tissue with mainly type 1 collagen fibers, and the inner layer is cellular layer or chondrogenic layer which has chondroblast in them, and these chondroblast will lay down new matrix. So the perichondrium has two layers, outer fibrous, inner cellular. Then the perichondrium is richly supplied by blood vessels which will provide nutrition to the cartilage. And articular hyaline cartilage and fibrocartilage are the two varieties of cartilage which are not covered by perichondrium. Thus, the cartilage is covered by perichondrium except articular hyaline and fibrocartilage. In these two varieties, perichondrium is not covering the cartilage. As we can see in the diagram, there are two types of cells in the cartilage, chondroblast and chondrocytes. So, the chondroblast are mitotically active cells. They are immature cells and they are present near the perichondrium, or we can say they are present in the chondrogenic layer of the perichondrium. So the chondroblast, if we compare it with the chondrocytes, they are smaller in size, they are flat and they are having irregular processes. So these are young cells which show branching process, and the chondroblast have the ability to produce ground substance and fibers. Then the second variety of cells is chondrocytes. So we can see in the figure also the chondrocytes are differentiated and mature cells, and they are mainly means their function is to provide nutrition by diffusion, then to maintain and repair the extracellular matrix. So if we compare it with the chondroblast, the chondrocytes are larger in size. Then we have seen that the chondroblasts are mitotically active, means these are active cells, chondroblast cells, whereas chondrocytes, these are resting cells, and as they are large, their diameter is about 40 micron and they have round euchromatic nucleus. The cytoplasm of chondrocytes is granular and it is more basophilic, and the chondrocytes are embedded within the intracellular matrix. So just to revise, chondroblasts, these are smaller and flat young immature cells which are mitotically dividing, and they are present in the chondrogenic layer of the perichondrium. They can produce ground substance and fibers, whereas chondrocytes, these are larger cells. These are in resting stage and their diameter is about 40 microns. They have euchromatic nucleus and the cytoplasm is basophilic, and where these chondrocytes are located, they are embedded within the intracellular matrix, and their main function is to provide nutrition and to maintain the extracellular matrix. There are four types of cartilages. So, first variety is cellular cartilage, which is present in embryonic life during the development of cartilage. Second variety is hyaline cartilage. It's mainly meant for support. Elastic cartilage is for flexibility. And the fourth variety of cartilage is fibrocartilage. It is meant for bearing weight. In embryonic life, during development of a cartilage, the fibrous mesodermal membrane gets transferred into cellular cartilage. So, the cellular cartilage is formed by chondroblast cells and there is minimum matrix. As the name suggests, cellular means more amount of cells are there and less amount of matrix is there, and the outer side of cellular cartilage is covered by perichondrium. In postnatal life, before the bone completely ossifies, cellular cartilage is present in resting zone of epiphysis. Means if we see the epiphyseal plate of the growing end of long bone, we can see the cellular cartilage in the resting zone of epiphysis. Second variety of cartilage is hyaline cartilage. The literal meaning of hyaline, hyalos, is glass-like. So hyaline cartilage appears glass-like because the matrix or the ground substance appears homogeneous. Various types of fibers are laden down in the ground substance, but they are not visible, and thus the ground substance appears homogeneous and it gives glass-like appearance. That's why it is known as hyaline cartilage. And hyaline cartilage is the most abundant variety of cartilage, and it is particularly seen in fetal skeleton. Then hyaline cartilage is also seen in articular cartilage. Now where this articular cartilage is present, the part of the bone which takes part in formation of joint is covered by articular cartilage. Then the nose also has hyaline cartilage in it. Then costal cartilage is the cartilage that connects ribs with the sternum. Then in the larynx we have cartilages. Few of them are hyaline and few of them are elastic cartilage. So the hyaline cartilages of larynx are thyroid, cricoid, base of arytenoid cartilage. Then we have C-shaped hyaline cartilage in trachea. Intermittent flips are also seen in the bronchi, and in the developing bone there is epiphyseal plate where we can see hyaline cartilage. Then what is the function of hyaline cartilage? As we can see in the respiratory tract, it is present in trachea and main bronchus. So it helps in maintaining patency of the airway. It prevents collapse of the airway and provides firmness. Then the costal cartilages, which connect the ribs with the sternum, provide support and protection to the thoracic viscera. We know within the thoracic cage there are two vital organs. There is a heart and a pair of lungs. So these are protected by the hyaline cartilage. Then the articular cartilage, which covers the part of bone which is forming joint, provides smooth surface for locomotion. The matrix of hyaline cartilage shows empty spaces called lacunae in which the chondrocytes are present. If we see the composition of the matrix of hyaline cartilage, it has 60 to 80% water, 15% collagen fibers, and 15% proteoglycans, glycoproteins, and 3 to 5% of cells. So if we see roughly the composition of hyaline cartilage, only 5% cells are there and 95% is the extracellular matrix. And if we see the fibers, they are mainly type 2 collagen fibers. 80% of the fibers are type 2 collagen fibers which are embedded within the ground substance. And again, it is very important for viva. Why the ground substance appears homogeneous or glass-like. There are so many fibers within it. Like you have previously seen the slide of loose areolar tissue, or there is fibrocartilage in the next slide. So there you can make out the collagen fibers. But in case of hyaline cartilage, though we have so many collagen fibers laden in the ground substance, why these are not visible histologically? So the reason is that collagen fibers in the hyaline cartilage have the same refractive index as that of ground substance. That's why ground substance appears homogeneous and glassy in appearance. And the composition of ground substance is mainly proteoglycans, and there are three varieties of proteoglycans present in hyaline cartilage: hyaluron, chondroitin sulfate, and keratin sulfate. Matrix, we have lacunae, and in these lacunae there are chondrocytes, and the chondrocytes produce sulfated proteoglycans. Depending on the concentration of sulfated proteoglycans, there will be variation in the staining of matrix, by which the matrix is divided in three zones. So the first zone is known as capsule or pericellular matrix. So we know that the chondrocytes are present in the lacunae, and immediately surrounding these chondrocytes there is a densely stained matrix that surrounds chondrocytes. And as it has more amount of sulfated proteoglycans, they have more staining, and they have more amount of type 6 collagen fibers. So we can see in this diagram also, the first zone is capsular matrix. Now immediately surrounding the capsular matrix there is territorial matrix. So as the territory in that region or in that zone, less amount of sulfated proteoglycans are there, so the territorial matrix will stain less than the capsular matrix. And the third zone is interterritorial matrix. So it surrounds the territorial matrix and it has less or least amount of sulfated proteoglycans, so they are lighter in stain, and they stain lighter with hematoxylin than territorial matrix. In previous two slides, we have seen the first component of hyaline cartilage, that is matrix. Now, the second important content or component of hyaline cartilage is cells. So, there are two varieties of cells, chondrocytes and chondroblasts. So, the chondrocytes divide and deposit matrix around them, and these chondrocytes occupy the lacunae in the matrix, and in living state we can see the chondrocytes are occupying entire lacunar spaces. And particularly in the hyaline cartilage, the identifying feature or the peculiarity of the hyaline cartilage is presence of cell nest or isogenous groups. So the chondrocytes divide to form daughter cells and they are surrounded by territorial matrix, and this is known as isogenous group or cell nest, which is peculiarity of hyaline cartilage. So what is cell nest or isogenous group? It is the chondrocytes that divide to form daughter cells. So two or more chondrocytes surrounded by territorial matrix forms the cell nest appearance. Now the second variety of cells is chondroblast. So we can see the chondroblast in the cellular layer of perichondrium. So these chondroblasts get trapped in the extracellular matrix, they lose their capacity of cell division and they are converted to chondrocytes. Hyaline cartilage is covered by a fibrovascular membrane known as perichondrium, which has outer fibrous and inner cellular layer. Hyaline cartilage is more prone to degeneration and calcification, and there are two clinical entities related to hyaline cartilage. First is osteoarthritis. So, osteoarthritis is a degenerative disease, and as the age advances, mostly seen by the age of 65 years. So we know that the wherever the joint is formed, the bone is covered by articular cartilage. So as the age advances, there are degenerative changes occurring in articular cartilage, and due to this there is chronic pain in the joints, particularly in those joints which bear weight, means those involved in weight transmission also, like hip joint and knee joint. And second clinical entity is rheumatoid arthritis. So it is autoimmune disease in which there is inflammation of joints. The joints have pain, there is swelling and warmth in small joints of the hand. So it occurs because the synovial membrane is thickened. Then conversion of hyaline cartilage to fibrocartilage is also one of the earliest sign of aging. Some of the textbooks have classified hyaline cartilage further into costal cartilage and articular cartilage. So costal cartilage is the one present between the ribs and sternum. So here the cells and the nuclei are larger in costal cartilage, and cartilage canals, which are commonly seen in the embryonic or developing cartilage, is also seen in costal cartilage, and it helps in respiratory movement and brings elasticity to the thoracic cage. Then the second type of hyaline cartilage is articular cartilage. Histologically, it has typical appearance. There are three zones in articular cartilage histologically, and it is present over the smooth surface of the bone, which takes part in formation of synovial joint. And we have discussed earlier that perichondrium is absent in articular cartilage. And there are three zones in the articular cartilage. The most superficial zone is tangential zone. Here the cells are small and flattened, as you can see in the diagram. Then the second zone is middle zone or transitional zone. Here the chondrocytes become spherical. Then the third zone is deep zone. Here the chondrocytes are arranged vertically. There is columnar arrangement of the chondrocytes. They are parallel to collagen fibers and perpendicular to the joint line. And the deepest layer is calcified zone. So the calcium is deposited in the matrix, and as soon as the calcium is deposited, the chondrocytes will not get nutrition and they die. So this zone, calcified zone, has integral role in securing cartilage to bone by anchoring collagen fibrils to deep zone. And here the cells are few in number, and the cells will enlarge, they become hypertrophic. And it as I told you earlier, that the deposition of matrix, due to deposition of matrix, the chondrocytes will not get nutrition. So here it receives nutrition from synovial fluid or vessels of the synovial membrane, or from the hypochondrial vessels near epiphysis. And the articular cartilage does not undergo calcification or ossification. So the third variety of cartilage is elastic cartilage. Elastic cartilage also has cells, mainly chondrocytes, then fibers, collagen, and abundant elastic fibers are there, and there is ground substance made up of proteoglycans. And elastic cartilage is also surrounded by perichondrium, like hyaline cartilage. And if we see the fibers in elastic cartilage, more amount of elastic fibers are there which have a branching tendency. They anastomose and they impart yellow color to the cartilage. And very few amount of type 2 collagen fibers are also there. And elastic fibers can be seen by using special stains like orsin or resource infused staining is used. And most important function of elastic cartilage is it is providing strong but flexible response to tension and compression. And here in this slide is of epiglottis. So we can see lining epithelium, lamina propria, some glands, and deep to that is the elastic cartilage. So epiglottis is an example of elastic cartilage. So in the central area we can see the elastic cartilages in the epiglottis. In the elastic cartilage, chondrocytes occupy lacunae within the matrix, and here the cells are bigger than the cells of hyaline cartilage. And chondrocytes are able to produce elastic fibers, collagen fibers, and ground substance. And the elastic fiber is also covered by perichondrium. In this flowchart, we can see the components of elastic cartilage. So mainly it has chondrocytes, uh, cells, and matrix has got more amount of elastic fibers, ground substances made up of proteoglycans and glycoproteins, and we also have covering of perichondrium over elastic cartilage. Elastic cartilage is located in the pin of external ear, then the walls of external acoustic meatus in epiglottis, corniculate and cuneiform. So epiglottis, corniculate and cuneiform are the three cartilages of larynx which are elastic in nature. And also we have elastic cartilage at the apical part or tip of the arytenoid cartilage of larynx and in the eustachian tube. Fourth type of cartilage is fibrocartilage. So fibrocartilage appears white in color, and in this, as we can see in the diagram, there are rows of chondrocytes embedded in the matrix, and matrix has abundant amount of thick bundles of collagen fibers. So here the fibers are mainly type 1 and type 2 collagen fibers, and if we see the arrangement of the collagen fibers, it is more like of dense regular connective tissue. So earlier we have seen the slide of tendon as example of dense regular connective tissue, and you might confuse fibrocartilage slide with the slide of tendon. So the important difference is that in fibrocartilage we have the chondrocytes embedded within the matrix having thick bundles of collagen fibers, and very few, some fibroblasts may also be present, whereas in the tendon there are wavy bundles of collagen fibers, and in between you can see the nuclei of the fibroblast. So in the fibrocartilage, there are more amount of collagen fibers, and chondrocytes lie either single or in isogenous groups, and mainly the chondrocytes are arranged in rows between the bundles of collagen fibers, and the chondrocytes lie in the empty spaces or lacunae of the matrix, and chondrocytes are able to produce matrix which mainly consist of proteoglycans and glycoproteins. Again, the point to remember is fibrocartilage is not covered by perichondrium. Fibrocartilage is located in the intervertebral disc, then pubic symphysis, menisci of knee joint, articular disc of sternoclavicular and temporomandibular joint, and in the glenoid labrum and acetabular labrum. And the function of fibrocartilage is that it helps in shock absorption. It can withstand compression and shearing forces. So this is particularly true for intervertebral disc and pubic symphysis. So these fibrocartilaginous tissue will help in shock absorption. Clinical significance of fibrocartilage is first is achondroplasia or dwarfism. So it is autosomal dominant genetic disorder where we have mutation in FGFR3 gene which causes defective protein synthesis. So the affected person has short legs and arms, and it has paternal transmission. Then chondrosarcoma. Now we use the term chondro for cartilage and sarcoma for any malignant tumor of connective tissue. So when there is malignant tumor of chondroblast characterized by excess secretion of matrix that usually involves the bones of axial skeleton is known as chondrosarcoma.